adenine, guanine, cytosine, and thymine. Not exactly words that slip off the tip of your tongue. But without them, you wouldn't have a tongue. But let's move on. These nitrogenous bases create the sequence that we think of as genetic code. Remembering how the bases form base pairs will be important as you learn more about DNA. Replication, transcription, etc. But we're getting ahead of ourselves. As a shortcut, biologists often refer to the nucleotides in DNA by just the first letters of their base names. A, G, C, and T. Sort of like rock stars that are known by a single name. In 1949, Erwin Chargaff discovered that when sorting out DNA, the relative amounts of A and T were almost always equal. And the same is true for G and C. In 1953, Watson and Crick figured out, with help from lots of friends, that hydrogen bonds could create a nearly perfect fit between nitrogenous bases along the center of two strands of DNA. In fact, these hydrogen bonds form between only certain base pairs, and remembering these base pairs is key to our understanding how the code in DNA is passed on. From Chargaff's data, it's not hard to figure out that guanine bonds with cytosine and adenine bonds with thymine. But how can we help ourselves remember the pairs if, say, we have a really important test we need to pass? There are only two possible pairs. A pairs with T, and C pairs with G. If we can remember just one pair, we can figure out the other one. Here's a trick I use to remember the pairs. If you list out the four letters, A, G, C, and T, and take a look at the letters, you see that G and C are two letters with similar shapes. This similarity is how I remember that G and C form one of the base pairs. Now that I've matched G to C, that leaves A and T. So I know that A and T go together to form a base pair. So this simple trick, noting the similar shape of the letters G and C, is how I remember how the four nitrogenous bases match up to form base pairs.